How do monoclonal antibodies use CD markers for lymphoma? Imagine a highly skilled immune system soldier that can be trained to seek out and destroy cancer cells with pinpoint accuracy. That's essentially what monoclonal antibodies do in lymphoma treatment. They are lab-made molecules designed to recognize specific proteins on the surface of lymphoma cells, called CD markers. These CD markers act like molecular addresses, helping the immune system identify which cells are malignant and need to be targeted. In many types of non-Hodgkin lymphoma, especially B-cell lymphomas, the cancer cells display certain CD markers such as CD20, CD19, CD22, or CD52. These proteins are involved in normal cell functions like growth and signaling, but in lymphoma cells, they are often overexpressed or uniquely present. Because these markers are relatively specific to the cancer cells, they serve as ideal targets for monoclonal antibody therapy. Monoclonal antibodies are engineered to bind tightly and specifically to these CD markers. For example, Rituximab is a well-known monoclonal antibody that targets CD20. When it binds to the CD20 protein on lymphoma cells, it flags them for destruction. This targeting allows the immune system to recognize and attack the cancer cells more effectively. Once attached, these antibodies can activate several immune mechanisms. They can recruit natural killer cells to kill the lymphoma cell through a process called antibody-dependent cellular cytotoxicity. They can also activate the complement system, a part of the immune response that leads to cell lysis or breaking apart. Some monoclonal antibodies can directly induce programmed cell death, called apoptosis, in the cancer cells. Others may block signals that help lymphoma cells survive, such as immune checkpoints like PD-1 or PD-L1, restoring immune activity against the tumor. Beyond traditional monoclonal antibodies, newer therapies also target CD markers in innovative ways. Bispecific T-cell engagers, or BiTEs, are designed to connect T cells directly to lymphoma cells by binding to CD20 on the cancer cell and CD3 on T cells, bringing them into close contact to promote killing. Antibody-drug conjugates attach chemotherapy drugs to antibodies targeting CD markers like CD79B, delivering potent toxins directly to the cancer cells while sparing normal tissue. Chimeric antigen receptor T-cell therapy involves modifying a patient's T cells to recognize CD19, creating a personalized immune attack that can be very effective. The impact of targeting CD markers has been profound. The introduction of anti-CD20 therapies like Rituximab has significantly improved survival rates and remission durations for many lymphoma patients. These treatments are often combined with chemotherapy to maximize their effect. However, some challenges remain. Cancer cells can sometimes lose or change the expression of CD markers, making them less visible to the immune system. Researchers are actively working to develop new antibodies and therapies that can overcome resistance and target other markers or immune pathways. In summary, monoclonal antibodies use CD markers as precise molecular addresses to guide the immune system in identifying and destroying lymphoma cells. This targeted approach has revolutionized lymphoma treatment, offering hope for more effective and less toxic therapies. Always remember, if you or someone you know is considering treatment options, it's essential to consult with healthcare professionals who can tailor therapies to individual needs and circumstances.